Descriptive analysis or univariate analysis of measurement of central tendency and dispersion. We are going to see that how to measure central tendency, dispersion for the univariate data. Under univariate data analysis, one popular question we are interested to ask is what is the typical value taken by the data set, or what the value around which the data are centered. To answer those questions, we have to measure the central tendency of the data. We already discussed central tendency can be measured by means of mean, median kind of measurement. When you discuss about mean, it is not defined for categorical variable. The mode used for measure of central tendency for categorical variable. Another univariate measurement is spread or measure of dispersion. This spread or measure of dispersion can be measured by means of range value, interquartile range, average deviation, variance or standard deviation. In order to measure the interquartile range or quartiles, we have to divide the data of sorted data. First, we have to start out the data in ascending order. Then we have to divide the data, the sorted data, into four equal parts or quartiles. Those values that makes those divides will be called first, second, and third quartiles respectively. Or otherwise, it can be called 25th, 50th, or 70th percentiles. After sorting out the data in ascending order, the first 25th percentage of the data values are called as 25th percentile, and the second part is called as 50th percentile or second quartiles, and third part is called as third quartiles or 75th percentile. This division will allow us to understand 25 percentage of data is taking what range of values, and 50% of the data is taking what range of value, and 75% of the data is taking what range of value, and remaining portion of the data of taking what range. So the range taken by the data set in four quartile we can understand by means of this quartile analysis. From the quartile analysis, we can find out interquartile range, which is nothing but difference between third and first quartile. This value will give us the range value actually. The first quartile is first 25% of the values, and third quartile is 75% of the values. So this difference will give you the range bound of the data set. So it will allow us to understand what ranges of the values the data is taking, which is also called as descriptive measurement or descriptive analysis. To do this descriptive analysis, we can use a single command in our environment, which is called as summary. As we are doing all analysis on iris dataset, we can do this descriptive analysis and central measurement and dispersion measurement on iris dataset in our environment. So this command summary of iris will help us to identify those descriptive analysis values or interquartile range values. After running this command summary iris, we can see that the summary of statistic or univariate analysis results. For example, if you take sample length, what is the minimum value taken by the sample length variable? You can visualize here, that is 4.3 centimeter. and the first quarter value 5.1. So 25 percentage of the sample length is within the value of 5.1 centimeter, and the median value is 5.8. That means 50% of the value, or 50% of the sample length is taking 5.8, or the mid value of sample length is 5.8. Mean value is 5.843. That means on the average, the sample length of the data set is taking 5.843 centimeter value. And the third quartile is 6.4. The maximum value of sample length is 7.9. So you can see that minimum value is 4.3, maximum value is 7.9. The range value taken by the sample length is displayed. Similarly, other value range, that is 25% of the data is taking what value you can observe from first quartile, and 50% of the data are taking what values that you can observe from the median value, and on the average what overall the sample length data is taking the value is you can observe from the mean value. So this is called univariate analysis, or descriptive analysis, or summary of statistic for the given data set. Similarly, other variables under the iris data, sample width, sample length, and um, petal length, petal width, you can visualize under the summary outcome.